Good morning, good afternoon, everyone. So as usual, this meeting will be recorded, and so we plan to post it on YouTube. So let us know if you want us to edit out something that occur during the meeting. So our agenda for today is to discuss what we went over last week, the details of a data release paper. So as we discussed previously, we are trying to organize the analysis phase that will follow the data collection that is ongoing at the Allen Institute. And to help this phase, we converge on the idea of making a data release paper that would contain some first order analysis of this data. And Sarah and Lucas put together a little bit of material to help lead the discussion, and also prepare a little bit of a paper scaffold to help organize this. So maybe I can start with showing a previous data paper that we made in the past. Does that sound good? Let me pull it up. So for previous publication with Colleen Gillon, we put together one of these. It's here. So this is a case where I'm going to pull the PDF so that you can see. Let me share it here. So the intent of this piece is to help document every aspects of a data set to facilitate data use, mostly data reuse, but in our case that will be data use. And this piece has several sections. First, you explain the experimental design with a rationale and the background for the study, and then any sort of information that is helpful to understand how the data was acquired. So in this particular case, it was chronic 2-photon imaging. Then there are data tables that explain, all of the experiments that were done with important metadata to understand the content of the data set. Then this is just explaining the stimulus of that experiment, along with behavior data, how it was processed. So you sort of help understand the extracted data that is available. And so this is, for instance, going over the data record to explain the entire content of each files that is shared. And then some first order analysis here. The stimulus at Gabor. So this is showing the responses of a number of cells, exemplary cells, to the stimulus. Same thing for a behavior, just to helping access this information. And then a measure of the quality of the recording. So this will be a SNR, amplitude, distribution plot of the units that were recorded in that session. And then there was cell matching done, and so this was documented in this piece. So it's a 12-page long manuscript. I don't think it's excessive in terms of work. So I took that piece, and let me share my other window. Where is that? Maybe here. Yeah. So I took that piece and I scaffold a document that is linked in the thread here. And so it lists the same thing for data release paper. So the background, we'll need to fill this in, the background and rationale, Probably will need a number of figures. I'm just tentatively listing some of these. So here, for instance, we could reuse the graphical abstract that we made for the review, or some version of it, and then reuse the experimental design figure. We already made these. And then so that would be the first with the background and rationale, and then there's the method. So experimental design will go in there, as well as the pre-processing, and just explain all of those things in depth as a normal method section. And then the data record. So that's the part that is probably different than a normal paper. We just explain, in great depth and there the data table will go. I think we already started the work with the Excel sheet that we shared. And then a technical validation. So that will be show of data quality, stability, reproducibility of certain things, and sanity check. So there we could put the temporal alignment check that we would do for the paper. And this list is completely tentative. I think this is what Sarah and Lucas were trying to build up. What are the figures that we would have in this piece? And some usage note in the end that would just document how one could use the data set in different directions that you could go. So this is a potential path to put this piece together. Are there any questions or comments, or do you think it's a good idea? I'm open to any ideas there. Oh, some of you are muted, I think. Or maybe it's me.
Microphone issue now it works.
Oh yeah, I can hear you now.
Perfect. Yeah. No, I was just saying this is super helpful. Also, kind of having the comparison to that other paper. This makes a lot of sense, and I agree. Now, I guess the discussion part is really on these extra figures, and
Yep.
maybe that's a good moment to pull up the poll result that Sarah.
Want to see that?
Yeah. Yeah, maybe we can use this bit as a basis. I mean, only 4 people participated, so it's
Yeah, we
not a
have a
representative
low.
probably, but gives us a bit an idea of kind of what are possibilities.
Yeah, and in my mind, it's also like a nice thing to sort of assign credit to all the people that help collect the data, and then any person among you that want to help out characterize the data set will also get another publication credit, which I think is very valuable. Because there's a lot of work. Any other comments, or like, should we discuss the content of the last figures, or what do you think?
I mean, to me, it seems that, I mean, this is just 4 responses, but there's some consensus that we should have at least receptive field mapping. And so if I look at the other paper that you just showed, Jerome, I think what any neuroscientist would want is some kind of a trial average, right? So just so you know that there's responses in there, and we all have seen pre-processing issues where we don't get a trial average because there's something misaligned about the timestamps or the snippets presentation. You want to see that that's all taken care of. So
That's
the
right.
receptive field mapping could do all that. We could also add what I can see the argument to show standard oddballs because that's the goal of this data. My concern here would be that if we show standard oddballs across all of these modalities, that may be a paper on its own, and it may be
That's
a
right.
higher paper than a data paper. So we could maybe go for that, and if there's nothing interesting there, add it. But I could see that would be our very next paper. It's the next low-hanging fruit to just compute the oddballs for all these different modalities and then make the paper that I think you envisioned, Jerome, where we say, hey, look, we go from synapse all the way to whole brain or entire networks of areas of what are oddballs doing, because it's the same stimuli, same paradigm, just different levels of measurement.
Yep. I mean, we also don't need to sort of characterize the standard oddballs in extensive depth, right? You could just highlight responses and then do not characterize the spatial temporal distribution across all the modalities, just to sort of as a primer. You could also do that.
Yeah, I also think that makes sense. One question I had was when we show these figures, do we focus on kind of showing this for some example units, or is it about highlighting kind of what's the quality or responsiveness also across all the recorded units? Kind of, do we still like stress more the statistics of this is how many responsive units we have, or look, these are 10 responsive units and they look very interesting, or something like this?
Well, I mean, it depends, because you could in the previous scientific data, there was like, for instance, measure of SNR. So you're sort of saying there are good units across all the probe, for instance, and in the number of units that are recorded across all the probes. So this is independent of the particular visual stimulus. So you're sort of supporting that the data quality is good and everything has been checked. As Alex said, if we dive into analysis of stimulus evoke response, that's starting to get into the science, which I think is could be beyond the scope of the paper. But it's an exercise we can play with as the piece is shaping, I would say. Farzana, you have questions?
Yes. First, I had missed this, sorry, but I do have some questions. So what do we, by contextual modulation and standard oddball and visual mismatch, are these questions, again, like more research questions, or are they more characterization? Because my understanding was that this paper is all about characterization. So that's why receptive field tuning, orientation tuning, those make sense to me. But then the last three parts, I wasn't sure what those are about.
Lucas, do you want to answer that? I think you'd be best.
Yeah, the question was if we want to highlight that there is some form of contextual modulation in the data that could be, for example, taking the oddball stimulus and comparing the oddball responses versus the control responses, because this is something that the stimulus design allows to do. And this might be just a kind of sort of a demonstration of what you can do with the data set. But this is really, really up for discussion if this makes sense, or if this is already too much scientific advance, and here it's just about sharing the data. But that could be applied to any oddball. Yeah, you're right. This could be, for example, standard oddball or the visual motor mismatch. So this was more generic, kind of, do we want to highlight what this paradigm can do, or do we just want to share the data, essentially?
So I guess what I'm learning is that you're saying the contextual modulation, which applies to standard visual motor, the idea is characterizing the response to the stimulus and to the oddball. That's how you say it, without getting too much into the interpretation of what these results mean. So it's more characterizing basic responses, which involves responses to the stimuli and oddballs. Is that how you're looking at it? And also, are we all on the same page that we want to keep this first paper more like just data characterization paper as opposed to answering a specific research question?
I'm not sure I understand your question, Farzana. Are you saying that you're
so what I'm saying is
do you have a preference?
I was thinking that this would be more of a data characterization, and then the papers that, but then again, it's not binary. Like data characterization could be also characterizing the response to the stimulus and oddball without getting very much into, like, without involving a modeling work,
Yeah.
without including. So it's a continuum. Like it depends what we define as research question and what we define as a survey and characterization.
Sure.
But
So in my
I
mind,
thought this is more survey characterization.
in my mind, this paper is intended to empower the next phase. So it's a documentation effort, maybe also code sharing effort to access all of the thing, and we will not aim to sort of answer any hypothesis that was stated in the analysis piece. Does that answer your question, Farzaneh?
Yes, I also saw Karim's hand is on. I'm sorry, I
Okay.
didn't want to take all the time. But with your definition, Jerome, then you would think that doing that standard oddball and visual model would make sense. So you think that's still within the range of.
I think that would be stretching it. I agree with you. But if, for instance, if you provide the code to start looking at this thing, like,
Yeah.
you know, and just highlight the potential of the data set in those directions, I think that makes sense. But you would not characterize it in depth with statistical tests and so on and so forth. Karim?
Yeah, so I think it's great to have a word out there about the existence of the data and what was done to collect it. So I 100% agree that this is timely. I'm a little bit uncertain on, packaging what we have just discussed right now without a much more clearer picture of what will go in the next sort of hypothesis validation paper or papers. So maybe to decide what should go into this data release paper right now should also somehow be tied to what will likely be the next step, as opposed to just leaving it vague at the moment, which is what the impression I'm getting right now. Maybe there were a lot more details on what will go into this hypothesis validation, more advanced analysis kind of next phase, but I still feel it's a little bit murky, at least in my mind.
Yeah. We could dive into this. I think Lucas had started to, make a figure with the different directions this could go. I wonder if because it's a longer conversation, right? Do you want to sort of bring the attention to that, Lucas, or?
Yeah, we could do that. Just that I mean, I'm not so, or I haven't thought deeply about the chunking these other pieces. Sarah had made an initial structuring. I mean, maybe just pull up the map as kind of maybe a resource we can then have a look at until another meeting to let me just share quickly to make this point. So this was Karim, this was the map. This was just an idea to how we could visually kind of structure this. And here I simply copy and pasted the things that Sarah has extracted from the community article. And so you're saying this, it would maybe make sense to figure out these in more detail so that we can kind of better assign what would be in the middle, which would be kind of the work packages related to data release versus some more advanced analysis that already pertain to some of these domains here, right?
Yeah, I think that definitely might help, will help. I think, these other colored analysis pipelines, ask very generic questions that were the focus of the bioRxiv article. But it wasn't clear to me that each one of these blocks would go and prove or disprove a certain hypothesis or set of hypotheses, which presumably will be the topic of a standalone paper, right? And so, one could think of, okay, maybe the bottom left set of analysis would just be focused on one hypothesis, let's say the dendritic hypothesis or the population one or whatever, so that when we have a clear picture of what each of these, peripheral blocks is intending to address, then we can decide on what will go into that center, ellipsoid basic analysis and data release paper.
I mean, Kareem, why can you clarify? Because I'm not sure. Why do you think? Because what we've described so far in the data release paper is a very shallow, thin piece that it's just meant to document the data tables, how the data is structured, with very little analysis into it. So why do you think it's coupled with the hypothesis in your mind?
Well, how the question is how little is little, right? If you're just doing basic analysis of receptive fields and saying, okay, hey, we're observing the same thing that everybody has observed for the past 50 years.
That's right.
But now the data is out there. So is that really the main message of the paper and nothing, no novelty, whatsoever, other than the slab
right.
2 design and that sort of thing?
That's
Is that the intention?
That's the intention.
Okay, so but the table or the survey is, as Farzana has suggested, is more involved, and so I'm not sure if the two are tied. If the data release paper is based on the outcome of the survey, or are these two separate things?
So the data release, I mean, it's up to decide what we put into it, but I would favor personally to not couple too much, the hypothesis with the data release paper, because for me, it's an empowering tool. Like we'll spend focused effort to document and make sure everything is clear for all of you to sort of go in different directions. And so if it becomes the case where the data release paper is too coupled to one particular hypothesis, that's not going to empower you. Because it's going to take time to review and so on and so forth. I would rather sort of make it as efficient as possible, and so that we can get it out. That would be my perspective.
Okay, that's great. That clarifies things. So let me ask, given that now you have all the template, right, the previously published paper to go off of, may I ask what was kind of what do you expect the reviewers' comments of this data release paper, given your experience in the previous paper? The novelty would be the mechanism of collecting the data, or is there something specific that we will highlight in this data release paper other than the fact that it was collected using the SLAB 2 microscope and other things?
I mean, the review were fairly straightforward. They were just making sure it was well documented. It's one of the easiest paper I had to push up.
Okay. That gives us hope that this is not going to take too much to get out. Yeah.
That's right. But yeah, you need to make sure that it's properly documented. I think the reviewer needs to see that the data set is impactful, so the background and rationale is important. Yeah.
Okay. Sure. That's enough. I just want to seek clarification and let Alex, take over.
Right. That's right. Alex?
Yeah, so related to that, I have a suggestion. So instead of when we say, why does this data exist? So what's your first item in this scaffolding you gave us, Jerome? Instead of making that about a predictive coding, what I would say is we make it about the multi-scale. So instead of the figure about, look, we take this from the review, there's this different predictive coding theories. Why don't we use this old figure where you have temporal scale, spatial scale, and coverage. This 3D figure where you see fMRI is here, EEG is there, and you see the trade-off for all these techniques. And then we show this whole area that we're covering, that we're going from synapse all the way to entire brain with perfect spatial and temporal resolution. And so we say that's what makes this data set interesting and unique. And don't talk about predictive coding too much at all. Like, of course, it's described in the paradigm and the stimulus, but thereby we can prevent any confusion in terms of that we're trying to, with this particular paper, address a scientific question. But we say if you're interested in data that has that unique, it's a first because step 2 is new, property of looking at the brain across spatial and temporal scales, then yes, here's your data.
Makes sense. That's definitely one of the strengths of the data. But that would be in figure 1, right? Stefan?
Wanted to mention that you might not even require something absolutely novel for a data paper, right? So to say, I'm a modeler. I scout the internet to see data sets and sometimes go on and try to ingest in some multimodal data to just go to try many things. So it's not, and there are resources which can be made where you do not need to say for the first time in the history of the universe, here we did this, which is often required for a lot of scientific papers. Right, so it is simply, but I do fully agree with you that simply describing a systematic data set across scale, I am actually not aware of things which have been done systematically across this. So could even that it is not a requirement, could be something which is new. But my recommendation would not be to insist on predictive coding, because then a reviewer would be as an, hey, you're saying predictive code, where is it? Right? You can mention that that is something which is, but if you make a dataset paper, you'd probably want to have that. There is another aspect for it, which I want to bring up, which is the fact that would be a good, there was somebody else who mentioned before a plan for the further analysis. And generally, I have lived a little bit in the physics community, and some of the colleagues in the astronomy community still have PTSD for data releases from observatories, which are coming because people end up competing and they are waiting when some data is released, and whoever gets some of the papers done first for that. So wanted to let you know that I don't think that the community is anywhere near the same, but at some point the community might move in that direction, that there is going to be a clock ticking for the people who want to do particular analyses or things like this, that you can't dump the data and expect 2 years later to publish a scientific paper on that. That it's not going to be instantaneous, but there better be a plan and there better be a way to try to think about that as to how you're going to reach that, not with no expectation. So my strong recommendation, knowing that community, would be release it, open everything like this, but have a plan for clearly who's going to try to tackle the next things in the future which you want there.
Yep. So should we, yes, Alex?
Yeah, I just want to say I agree with all of that. I'm just a little less nervous about scooping because, if anything, it seems so far the experience in this project is that, I mean, the data is already out there, right? But if anything, we're getting too little analysis and too little participation. So I think that what I'm learning is there's very few people who actually have the time and salary and everything that they can sit down, learn a complex data set and do that. Now, I agree that some modelers might be doing that, but they probably do this in a particular context, maybe of their model. But like the stuff that kind of is now already, you could go and write a paper and take what we do and study predictive coding. And a lot of us, we even show up every week and we just don't have the time to even start doing that. So it seems to me that, and also with some of the other OpenScope data, it seems that there's very little pickup. So yeah, I share all of this because I'm from the old times and we wouldn't even release our data because we were so worried about scooping. But I'm blown away by how little that seems to be an issue. So I'm for full transparency also.
Yeah, I mean, making the plan for analysis is useful no matter what. I think, so we should do it. But I'm hearing this consensus with putting together this data release piece. Is there any, given the clarification we made, are there any concern or? Okay. And Sarah put together an Excel sheet. Maybe it's worth bringing that up.
Jerome, just very quick question. So when you wrote this other data nature data paper, how is the code handled? Obviously, it's about the data as a package, but then do they ask that you also have a GitHub repo or that you have a Python notebook or something? That is there code release? How does this usually work with that journal?
Yeah, this was a few years ago. It's probably changed since, but yeah, you can have a Git repository along with it. I think it's will be better to do that. I don't. It wasn't too complicated in my experience, but for me, essentially, you will need like the Dendy set. That's what we had. The Dendy set that are well documented and released with DOI. There was a little bit of a chicken and egg problem at the time because it was very early where we had to get the DOI of the data release and the DOI of the paper, and so we didn't know which one first. And because the DOI of the Dendy was referring the papers, it was just a bit of a chicken and the egg problem. That's the only issue I had at the time.
So there are no particular
yeah.
requirements for the code, for example, how commented it is, or that it's a particular repo, like the Allen repo, because it's more institutional, was not just some random person's repo that could get pulled in the future. Or the journal doesn't care. I'm just curious.
Oh, I don't recollect. We should check so there. I mean, scientific data is under the Nature journal portfolio. I think eLife also accept this resource paper. And they might have different requirements. We could look at the different options for these. Personally, I don't really care as long as it's out there and open access. That's all I care about. You might have more preference, but
no, I just wanted to avoid that if we start doing coding in a certain way and then we have to change everything later. So if there are requirements, maybe that we know right away so we can write our code accordingly. I'm always surprised that there's so much emphasis on the data, but that there's almost no emphasis on the code, but that is often the crucial part to even make sense of the data. Maybe in the days of vibe coding, you don't need that anymore. But in the old days, for example, I've tried data sets from the internet online. I've given up because there was no supporting code or the code was broken. And then there was no way of making sense of the data, actually, even if you get all of the details in the paper. So
that's right. I'm just trying to pull the Excel sheet. Thought Sarah made one. Do you remember, Lucas?
You mean the kind of the overview of these analyses?
I thought she made an Excel sheet with.
Yeah, that was on. I think it's here. I post here there. My post.
Is it this one?
Yeah, exactly.
Oh, do you want to discuss that?
Yeah, I mean we could. This is I guess about these additional analysis that we want to put in. So here I just collected ideas really as kind of to clarify a bit what we meant with the points in the poll. But Yeah, I think from what I heard, there's a bit of a consensus that kind of a receptive field analysis would be good, just to show that we, stimuli were presented at the right spot and so on. Whereas these later points, so here in cell 9 and 10, this dynamics and tuning, responsiveness of a session, or contextual modulation, these are things that go a bit beyond. And I also was the only person ticking those, and I would also be completely fine dropping those if we really want to constrain it on the data. So I think this is all fine. Maybe an open point for the receptive field analysis. I think one thing that is interesting in the data set is that we include the zebra. So I was wondering if we should include kind of an example analysis of the zebra stimuli as well. Maybe that we could, in one figure, where we plot the kind of basic Gabor patches, receptive field analysis, and then next to it with the zebra, because it's really potentially tracking different units and characterizing their visual properties or so. There would be suggestions.
It makes sense to me. I mean, you're also sort of providing how to analyze the stimulus that is a little bit different than usual. So it's setting up the foundations for using it to characterize. So that makes sense to me. One thing that we might want to do is maybe create a list of panels, right? Like, so as you said, there might be a number of panels we want to include in this piece, and find an owner for each one of those panel, so that, well, first, if you want to be a co-author of this piece, this is the way to get obvious credit for contributions. And then also it helps us build the piece, because I think there's going to be panels about describing the experiments, panel about describing the multimodalities, like, panels about how the data was collected. And so I feel like we can create a lot of different pieces that are built in parallel with people across the group. So maybe we should do that. How do we do that? So should I start to seed this, and then and then people so could add ideas of panel they want to build for this piece?
Yeah.
Who is interested in contributing to this data release paper in the audience right now? I'm just curious. Alex, so I'm seeing the raise your hands if you want. I think that'll be easier. Farzaneh, Lucas, Kareem, Alex, Marcel. Okay. Looking at the whole group. Okay. That's good. So maybe what we could do is for next week, edit this Excel sheet, and then each line is will be either a paragraph or a panel on a figure, and then everybody can add what they think they would like to contribute. Does that make sense?
Jerome, maybe we can work on a rough timeline that you have in mind. I mean, we're still also collecting data. So shall we start on this while the data is being collected? And do we have a rough target date in mind where we should, see that we get this all done?
For me, the ideal case is the paper is written, packaged by June.
That's ambitious. Okay, cool.
Because we are all working to finish there's a little bit of a unknown about currently the data collection is going smoothly. Last week we I mean, Carter and I and others started to, Made and others started to push up a lot of data on CodeOcean. So it's actually now sitting on OpenBucket. We're going to give you access soon. But there's tons of data that is going up. The NeuralPixel is making progress. I think in the next few weeks, we'll have finished the first round. We're going to review the data set next week with Severin and others to finalize the last bits. 2Photon is also going smoothly. So we'll likely be done with the neurophysiology data collection in early spring as we plan. The things that I'm a little unknown about is the brain reconstruction. So every brain for NeuroPixel, we reconstruct to see the probe track. And there's a bit of a backlog in the imaging platform that help us do that. And so it's possible that we'll be late for the paper. But that will be the last beat, one of the last panels. So, for instance, in that paper, there will be a beautiful panel that shows the probe track across all of the experiments for NeuralPixels so that you see where the experiment are. And making that panel might be the last beat. But yeah, that's the target I had in mind, is just to finish it up early so that you guys have all you need to analyze it in more depth.
I have a
It looks to
question,
me that
Gerald.
Yes, Maddie.
Oh, sorry. Thank you. Are we planning to include the voltage imaging with ASAP 7 for this release? Because I know that we're still collecting data and no one has started working with the data.
Yeah. When we haven't shared it.
Yeah, that exactly.
I think that would be the idea. But
Okay.
for the data release paper, it's not about analyzing in depth. It's just characterizing making sure that data integrity is good. But as you said, yeah, we need to make sure that it's as part of the last week push, we actually push up some voltage imaging onto the cloud, and so we'll need to give you access to this.
Oh, amazing. Okay, it's good to know the deadlines then, because I'm still working on the voltage imaging. Okay, good.
That's right. Deadline is a tool to help everyone work towards the goal, so we can adjust as needed.
No, it's good. There's motivation.
That's right. So for next week, we share this Excel sheet. I'm gonna add rows that, to my understanding of what will be helpful. It will be great if each one of the person would raise their hand, also edit the sheet and had their thoughts on what they want to contribute to it. Any other questions?
I mean, it looks to me like the bulk of the actual paper needs to be written by the Allen team, right? Because it's a lot about the methods and whatever, where we might not have all the details.
I think a big part of the methods will be written by us. That's right. And that's okay. Because this is essentially it's something we need to do anyway to help you guys. And so this way we do it on once with feedback from all of you. You'll be the reviewers. And then we make sure it's well documented.
So I assume we're gonna do this again, and we have a Google Doc maybe that is shared between everybody, that where everybody can see what progress is and edit and comment and so on.
You have it already.
That's gonna be the paper. Okay, cool.
Right? No? But we need to agree on the figure panels first, maybe, because then everybody will write on different parts. But I don't underestimate the characterizations and all the work that you guys could do, making sure the data is properly formatted and so on. I think that's really important, and that's a central component of the data release paper. So, because the code that you guys write for that will be part of the paper, right? Okay. Marcel?
Yeah, one question. So when you speak about panels, do you mean something like a figure sketch? Or what exactly do you mean by panel?
Well, say for instance, we want to include an analysis of zebra, right? And so let's say we have a figure about receptive field analysis in the dataset. How to do that? There could be one panel about zebra that shows the receptive field you see using zebra analysis. So for me, a figure in a paper is telling you a story about a particular thing that you're trying to highlight, and then there are multi-panels that support that story. And so for me, a workable piece is like a panel.
Mm-hmm.
It's a chunk of work.
Okay, I see. Cool.
Marcel's idea, I wonder if in addition to this Google Doc, we can have a Google slide where we can do what Marcel suggested, that maybe all of us are like, here's a cartoon, or just you make up a square and in text, you write, this is the figure that I want to contribute, and, you know, a sub-figure panel. And so that might help us be organized. And then actually we can then cut and paste them from when we have the figures in that Google slide, because Google Slide and Google Docs works nicely. You can take a Google slide and paste it into Google Doc and then update it and keep it updated. And so you can keep those two together, and you have your figures always with the text. And it might also keep us organized maybe a little bit.
I see. So a Google Sheet for the list of items, a Google Doc for the text, and a Google Slides for the figure. Is that right?
Yeah, when I work with co-authors, we often have a Google Doc, and then we have a Google Slide, and all the figures are slides in the Google Slide. And you can cut and paste in your Google Doc, and then when you update the slide, you just hit update, and then they update in the document as well.
That's right. That's what we did for the review piece, and eventually we use Google Slide.
Oh.
Okay. So we'll plan to discuss that next week. This briefing, the Google Sheet, the Google Doc, and the Google Slide, and sort of solidify the plan for the data release paper.
Yeah, so for example, one question I have is, should we have every figure that we make, all three modalities next to each other? Should we always have SLAP2, EFIS, Opto, for example? Like these are decisions we can make before we get started on writing the code and analyzing.
That's right. That's right. I'm not sure. Like, it depends. Certain things are easier to analyze in one modalities than others. But there should be a figure that show the multimodal aspect, right? I like, maybe it depends. What's the story we want to tell? But there definitely should be a quick figure that show the three modalities with one stimulus. But maybe we should choose which one. Because, for instance, receptive fields in OFIS look different than receptive field in EFIS. And SLAP2 we haven't looked too much, but yeah. Stefan?
I actually think that if the point would be to include all these modalities, having these side by side, the three modalities, all the same analyses, different or not, whatever blemishes there are there, that would be kind of like the essential thing to have. That I would strongly recommend that to actually be the organizational principle around how you build everything. In terms of the figures, right, that you're going to have. And sometimes it's also easier when you've got a mental model as you go through figures when they have the same overall structure. So if the schema is the same, people have an easier time and you can follow through that. But at least in my mind, I would love to read a paper where all the analyses are done in parallel on all the three modalities, and some would not be very good, some would be better, but that is exactly the thing which would bring humongous value to that.
That's right. That's right. We could do that. But does mean a level of alignment. Yeah, but it's fine.
I think that would be that is the thing which actually proves the alignment between the data, and that's the thing which pushes. And if you find that something is totally off, that is the thing which would raise a flag or understand, or they might be totally off and there might be a very good reason for that and you can explain it, or they might be totally off because there has been a bug somewhere, or right. So it's not on having this in parallel is going to add, I think that is the main power here.
That's right. That's right. I was on a course trying for the study. We're already into next week list of panels. It's good. Any other comments? Sounds like we have a plan. Okay. Well, it was such a wonderful, lively discussion. I really thank everyone for your contribution. Let's meet next week.
See you. Bye.